So John received his PhD from Brandeis, and then he went to Stanford, where he was a postdoc with Dave Hoopness, and he worked with on the Drosophila system. And then for his first job, he went to Cornell as assistant professor, and that's where John and I overlapped. I was a postdoc at Stanford when he was a postdoc, and then I left and went to Cornell, and he followed. All right, so we've been, I didn't meet, but it was temporally, I was there first. So anyway, so we've been friends and colleagues for more than 3 decades. So I'm really very happy that he's here. He's one of the most valued colleagues at Cornell. He's one of those people who really care about the whole program. He was one of the faculty that attends all the PG seminars when he's in town, and he's in town a lot. And he really contributes to a lot to the well-being of the department. Now, John has a really stellar career. He always pushes the technology to meet new challenges in new problems. And when he first started at Cornell, he was looking at a promoter regulation of transcription, and he used the heat-shock gene as his model. And he found something that was, initially everybody thought it was just something special to heat-shock genes. Heat-shock genes are genes that are activated upon shift to high temperature. So he was looking at how look at the promoter activity there. And contrary to what most people believed at that time, that occupancy of the RNA polymerase at promoter was the regulatory step. But it turns out that wasn't it. It was something that the RNA polymerases were already engaged, and that they actually make a little transcript. They were just arrested, they were paused. And the regulatory step was actually the release of the RNA polymerase from the pause. And he started from this one gene, and now it turns out it is universal, it's global, and that's what he's gonna tell us. So welcome, John. And
thank you, Beck, and very nice introduction, thank you. It's been great to have Beck as a colleague for all these, and close friend all these years. And you are very, very lucky to have her here. And Henry. Okay, and we're sad. So thank you for the invitation to come here. This is really a terrific location you have here. The architecture is just wonderful. And what I really like about all of Hong Kong is the way you preserve the way the natural beauty is preserved as well. So you don't have buildings just overpowering these beautiful hills and islands and other things. So really doing the right thing here. Really beautiful. So as I think many of you know, higher organisms have evolved sophisticated mechanisms for regulating their 20,000 or so genes in response to a spectrum of developmental cues that create us, as well as the nutritional and environmental cues that maintain us. And a primary and major part of this regulation is at the level of transcription, that is deciding whether to make an RNA from genes. And the specificity of this regulation, how do you pick out the particular genes out of these 20,000 to turn on or turn off? This all relies very heavily on a large number of DNA sequence elements. And there's a class of elements that are associated with the core promoter, that is the place where RNA polymerase binds along with the help of general transcription factors to initiate transcription. There are also elements further upstream to which regulatory factors can bind. These regulatory factors are proteins that have usually domains that are a chemical complement of the DNA that they recognize to provide the specificity for interacting with a particular sequence. Sometimes these elements here can be quite some distance from the gene they regulate. They can be, well, actually across campus here, either upstream or downstream, and still act on this particular gene. And these collections of elements to which these factors bind are called enhancers. Now, these regulatory factors can sometimes interact directly with the polymerase and to recruit or increase the activity of the RNA polymerase. Alternatively, or more often actually, there are co-regulators involved that, that interact with the regulatory factors, domains on the regulatory factors, and they then in turn affect the recruitment or activity of RNA polymerase. But they often do more than that. They're multi-subunit complexes, as I've tried to illustrate from this cloud-like structure here, where there are subunits that can then interact also with other things. And these, well, let me first say that these co-regulators come in several flavors. There's a mediator, a very multi-subunit complex that interacts with a host of transcription factors and polymerase. There are histone acetyltransferases, or HATs, and there are chromatin remodelers that burn ATP and can push nucleosomes around. And so there are subunits in this complex that can modify the histones or move them around and make the template here a better or worse place for transcription to take place. So finally, there is also some mysterious elements called insulators that can block the activity of these enhancers and in a sense restrict or punctuate the genome so as to restrict the enhancers so that they don't affect the wrong genes, so that they target genes that they are supposed to target. Okay, now to study this system, we decided many, many years ago to focus on, as Bick pointed out, the heat shock genes, in particular the heat shock genes of Drosophila. And we chose this system for several reasons. One, these genes are robustly activated. They're induced over 100-fold. So even if you're a sloppy scientist, you can do good science. You can make these measurements and see what the effect of mutations are. Response is rapid and synchronous. So within seconds, you can begin seeing factors be recruited. You can watch polymerase as it moves through the genes. And knowing where the factors are and where polymerase is gives you some clues as to how these genes are working mechanistically and how they're regulated mechanistically, and then be tested. The principles and underlying mechanisms proving to be general, much more general than I had imagined. And finally, in Drosophila, we have polytene chromosomes that provide amplified signals and resolution. Those of you who don't know polytene chromosomes, these are special chromosomes. They're interphase chromatin, so the chromosomes are extended and active, but they become visible because they're rounds of replication without cell division. So you can have 1,000 copies of these chromosomes aligned, and now all of a sudden these fuzzy chromosomes become highly visible and highly banded. And this lets you have a strong signal at a gene if you're trying to probe for a particular sequence or a particular protein. And you now have, because they're interphase and extended, they're not like metaphase chromosomes, so you can have resolution as well. And this, just show you an old example of how these can be used. Here is a chromosome, a small piece of the genome. It represents less than 2% of all the polytene bands and polytene chromosome in the cell, spread out here. And these contain the heat shock genes, the major heat shock genes, the HSP70 genes, with 4 copies at this 87C locus, 2 at the 87A locus. And upon a heat shock, within 10 minutes you see, and these are at the same magnification, you see this dramatic decondensation at these loci as they become activated. And we can see that they're activated because we can stain these with the machine RNA polymerase, which transcribes at these heat shock genes. So this can be repeated with a variety. You can stain with antibodies to a variety of proteins, and you can also do is this technique of chromatin IP, where you immunoprecipitate particular proteins and look to see what DNA co-precipitates with them. And using those kinds of assays, we can get a picture of what the gene looks like before it's activated and what the gene looks like after it's activated, right? And so before it's activated, and I'm only going to show you the key players. It's somewhat more complicated or sophisticated than this, but the key players are all we need to deal with today. There's a factor called GAGA factor that binds GAGA sequences, GAGA sequences, and that is critical in opening this whole chromatin structure. That's moving the nucleosomes out of the way. These are the nucleosomes over here. There's an RNA polymerase on this uninduced gene that is before you heat shock that's associated with the promoter, as Bick mentioned, and it's made somewhere between 20 and 50 bases of RNA and has come to a pause. And that pause is stabilized by protein complexes of NELF and DSIF. These are two protein complexes that bind this early elongation form of RNA polymerase that has already initiated and begun making RNA. Every 10 minutes or so, this polymerase escapes or is released into productive elongation and gives rise to an RNA. And this gene these genes are expressed at some level, but they get expressed much higher rate, as I mentioned, over 100-fold higher rate upon heat shock. Upon heat shock, the master regulatory protein, HSF, which was a monomer, now trimerizes and becomes activated and binds with high affinity to its targets in front of many of the major heat shock genes. And this HSF, we think, indirectly recruits, but it's critical for recruiting, a kinase called PTFB, something that David Price has discovered and studied intensively. This is a kinase that works to phosphorylate the components of the pause complex. It phosphorylates the polymerase, DSIF, NELF, and then changes the properties of that complex, making DSIF, transforming it from a pausing factor to a critical elongation factor that helps speed polymerase through the gene and through any obstacles there, like nucleosomes and other things. But in any case, it helps elongation. NELF, that complex, pausing complex, leaves, and polymerase itself becomes heavily phosphorylated on this tail that it possesses on its largest subunit. That has a repeated heptapeptide, and that's a target for intensive phosphorylation. It's also a target then for binding of a lot of factors, and we have factors binding, and polymerase creates a whole entourage of proteins that accompany it as it transcribes through genes. Some of these proteins involved in processing the RNA into a proper spliced and capped product. Okay, so over the years, we've emphasized an embarrassingly simple strategy to study the process of transcription. We decided, even though we were in a department that really strongly valued traditional biochemistry, we decided to do dirty biochemistry, in a sense. We decided to look at live cells and simply observe them, then perturb them, and then reobserve them soon after. So it's very, very simple, except to really get information and begin to approach mechanistic information, not quite the information that you heard about Seth a few minutes ago, but information nonetheless. To get good, useful information, we have to look at very high resolution. And so we want to be able to look at genes and genomes and RNA polymerase and transcription factors at the highest possible spatial and temporal resolution. We'd like to be able to look at base pair resolution at factors in vivo. We'd like to be able to watch things as they change in a time frame of seconds, if at all possible, or at least minutes. And we can do this optically, gene by gene, we've done this making use of polytene chromosomes, as you'll see. And you can do this also biochemically, looking at targeted genes with various approaches that let you cross-link proteins to DNA and otherwise modify DNA to map what's happening. But more, in the past several years, we've turned to genome-wide assays because you can get the same kinds of information, sometimes better information, by looking at the entire genome rather than looking at individual genes. And you can even do this more cheaply, and at least certainly on a gene-by-gene basis, but even, you can even begin to approach the cost of doing single-gene experiments with genome-wide data. And also, this provides us statistics, so we're having all of these genes looked at once, and also ways of testing the generality of the models that one creates by looking at individual genes. So we can then perturb, and there you all know of a variety of ways of perturbing various factors. You can make mutants, you can do RNAi to deplete. There are drugs, and you've heard about some of those from Seth. Today that'll inhibit specific proteins or inactive sites in particular proteins. And we've been also working on RNA aptamers, another strategy, about 15 years or so, trying to generate RNAs that have high affinity for various targets. And we can then look at these but unfortunately I won't be talking about RNA aptamers today. I'm not quite ready for prime time. But we will talk about these other reagents, other strategies for perturbation. And then you can look at these as soon as possible to look at the primary effects. Okay, so today's lecture I'm going to do in 3 parts. First, I'm going to, first we'll cover the background and a summary of published studies, and I'm going to go through this in a fairly quick way. And then the last 2 will be dealing with unpublished results. So let's start with the first part, which is promoter proximal pausing, which is a critical step in the regulation of a majority of what we think Drosophila mammalian genes. At least it's a rate-limiting step in many of those, and it's been to be regulated in others. So in principle, to understand how transcription regulation works mechanistically, we need to know all the factors and all the great biochemistry that's been done to purify factors, really critical in all of this, done by a variety of labs. But we also need to know what is the rate-limiting step to understand how these regulatory factors operate. And so in principle, if we look at a transcription cycle, any of many steps could be regulated. We could be regulating the opening of chromatin, pre-initiation complex assembly, the initiation process itself, promoter proximal pausing, escape from the pause, productive elongation, termination, and then eventually recycling of the polymerase. And there's been a lot of attention in these first steps, that is the opening of chromatin and the recruitment of polymerase and the initiation of polymerase. But we were forced to consider steps later on from our studies of Drosophila heat shock genes because polymerase was already associated with the promoter before we induced. And so we've been focusing on promoter proximal pausing and the escape from pausing in a lot of our efforts. And this is steps in early elongation. Now, what was the evidence that led us to worry, start being obsessed with pause polymerase? Well, first there was experiments done by one of my early grad students, Dave Gilmore. It was a UV chip experiment. We didn't call it UV chip then. We gave it a lousy, boring name of cross-linking in vivo, ... Nobody remembered it. When you create a new technique, give it a good name. That's my advice. Anyway, now we call it UV chip because somebody else invented the word chip, Dave Alice. Chromatin immunoprecipitation, except instead of using formaldehyde as a cross-linker, as people normally do, back then we used UV light and it was a zero-length cross-linker. That showed us, and we're using RNA polymerase to develop this. We're looking at heat shock and non-heat shock cases. In heat shock, we finally got this working. We could see polymerase over the entire gene, and in uninduced cells, before heat shock, we still saw a background, and when we mapped that, it mapped to the promoter region. And we quantified the amount of polymerase, and it turned out to be present at about 1 polymerase per gene. So this was something we could not ignore. What was the status of that polymerase? Well, we did, Anne Rugview did nuclear run-on experiments where we isolate nuclei, and under conditions where we strip away a lot of chromosomal proteins and allow those polymerases to transcribe, but we prevent any new initiations, we saw strong signals from that very 5' end. So that polymerase was not only bound, but it was engaged in transcription, okay? Now, how far had it gone? Well, another student then decided to map this precisely using chain terminating nucleotides and labeled nucleotides in various combinations, and analyzing the sizes of those labeled RNAs, and found that for the HSP70 gene, the major heat shock gene, the RNAs were between 20 and 40 bases. That means that polymerase had moved 20 to 40 bases, so we could map with base pair resolution where this was in vivo. And then this was complemented by permanganate mapping. It's a chemical you can add to cells, 1-minute treatment in intact cells, and it'll seek out and modify Ts that are in single-stranded DNA preferentially. So modify Ts, you can then break those modified bases and then run them on a sequencing gel. More complicated than that, but basically you can see precisely where those Ts are that are hypersensitive, and that told us that this bubble structure was indeed there, and that's characteristic of an RNA polymerase elongation complex. So all of this data pointed to the fact that there was a polymerase there, about 1 per gene, before the gene was heat shock activated. Now, there were studies by Grudine's lab and Ike's lab that showed similar Pol2 associations for c-Myc and c-Fos in humans. We had seen it also for several Drosophila genes. And that was, but in the early 90s, there was a real emphasis on studying yeast, which is a beautiful system, yeast Saccharomyces cerevisiae in transcription studies. We learned a lot from that, but it didn't have pausing. So people forgot about this largely, except us. And I was happy to, no, there were actually several people who were still thinking about it, but the emphasis turned to other kinds of mechanisms. And so, but in 2005, people began publishing chip data, these immunoprecipitation experiments using RNA polymerase to see where RNA polymerase is genome-wide, and it was showing up, piling up on promoters in mammalian cells. And here's an example of this from Bing Ren's lab, published in Nature in 2005. And here is the distribution of polymerase on this highly transcribed ribosomal protein gene. There's a peak of polymerase right around the promoter. You expect more polymerase on the gene, but it's actually easier to detect the polymerase around the start site here. And of course, there were other studies from Karen Edelman's lab and Mike Levine and Rick Young showing that they could see this as well in these chip experiments. We wondered whether these polymerases that are there on promoters were pre-initiation complexes, as was hypothesized in this paper, or could they be the pause polymerase like HSP70 that we saw in Drosophila? So we decided to try to contribute to this argument by developing a methodology called GRO-seq, Global Run-On Sequencing. That is a nuclear run-on experiment where we look at all the polymerases in the nucleus and ask where they are. And the way it works is the following. It's developed by Leighton Core in my lab, another graduate student. We allow the polymerases to run on. We isolate nuclei. And then we ask, where are all the polymerases? So we allow them to run on after treating the nuclei such that every polymerase sort of runs on the same way. The pause polymerases and elongating polymerases all will transcribe in a very similar manner. And we did this in the presence of a bromo-U TP, affinity tag. In the past, we had been using radioactivity. Radioactivities, even with single gene experiments, will get you in trouble with the radiation safety department when you use it at the level we were using. And we almost lost our license many times. Students were terrified of this. But then the bromo-U is a nice affinity, clean tag, no radiation, and we could incorporate multiple PRU-TPs into the nascent RNA, and then isolate that. We run on for a defined amount of time, then we fragment the RNA so that we can then map precisely where the polymerase was, fairly precisely. So the RNA is fragmented after this labeling, and then we do ligation-mediated PCR negation, and then we do PCR, RT and PCR to amplify that product, and then it's put through high throughput sequencing. And here's a case of a modestly expressed gene. This is one of the mediator complexes, and here's what the pattern looks like. If you have, here's the pause. We see this striking peak at the promoter, pause polymerase, and we see transcription through the gene. We also notice, so we were delighted to see this. We did this in the very same human cell line that Bing Ren had done made his observations by the chip experiment I just described, and indeed there was peaks, and we were this is one of the eureka moments in the where you're really happy in the lab. Somebody was asking me about that at lunch. There are those moments that you treasure. Start getting a data set back, and you're looking at your favorite gene, then the next favorite gene, and the next, and yeah, it's pausing, just like HSP7. I was excited anyway. I think Leighton was as well. And so anyway, you detect, this technique detects all of these, detects RNAs regardless of the polymerase or the stability of the RNA. You're looking at the nascent RNA. And so if you now look genome-wide and you plot all of the polymerase density on genes, starting from their transcription start site here, and with lining up all the genes left to right here, you can see the polymerase density is high, and polymerase is paused on strong level of pileup at plus 50, just where we'd expect. Surprisingly, and this was a shock too, is that there was a polymerase in the opposite direction that appeared to be paused. Even when there was no gene here, we subtracted out all the cases where there's real genes going in the other direction, but there's transcription going in the other direction in many of the cases. And about 80% of the promoters produce detectable divergent transcription. Often it can be stronger than the transcription that we see in the direction of the gene. So that was useful. One other technology, since this is the University of Science and Technology, right? I wanted to get one more technology point, and that you can map the 5' ends in nascent RNA very sensitively by an adaptation of this methodology. And so when you isolate nuclei and do run-ons and get nascent RNA, these RNAs are capped very quickly. As soon as the polymerase gets to plus 20 or so, the RNAs are capped. You can then enrich for the affinity tags and then do degradation of any RNAs that are not capped with a couple of enzymes. And then you can take the capped RNAs that are only from the 5' end, right, and remove the cap and then put a linker on that end and then sequence from that end and determine where the 5' end of the genes are. And here again, we're looking here at the standard GRO-seq patterns of polymerase density, but now if we do this GRO-cap method, we get rid of all of the other RNAs and we can just define where the transcription is starting. And this turned out to be extremely a sensitive way of identifying not only promoters, but also enhancers. Remember these enhancers, these things that work at long distances? Well, they are also transcribed. All of these papers support the idea that there is transcription of RNAs called eRNAs, enhancer RNAs, at known enhancers that are active. And we looked at that with this methodology. And I'm not going to take you through all of that study, but if you're interested, you can read this paper here in Nature Genetics. But what we found is that promoters have this polymerase that's paused in both directions and on genes. So we have, and they're typically separated on average by about 110 bases. There are factors that bind in between. They have the general transcription factors. And enhancers have the same architecture of initiation and pausing, and you see polymerases pausing at both directions. And they have EBP and general factors, and they have activating other kinds of regulatory factors in between. And the regulatory factors are fairly similar, though not identical, between promoters and enhancers. There's a striking similarity by what the structure of a promoter is and the structure of an enhancer. Now, the difference with promoters, of course, is that these polymerases that are transcribing a coding gene give rise to stable RNA, and that is largely because the RNA has evolved away sequences that render it unstable, particularly these polyadenylation cleavage sites that emerge in sharp, nicely documented. It's critical. We agree with that, and you get rid of these sequences that do cleavage and polyadenylation prematurely, and you stabilize the resulting RNA as well. And I won't go through that mechanism, but and it's not well defined yet, but it just happens. In this case, we have DNA sequences that run into these destabilizing. In this case, we have this polymerase making sequences that destabilize. Likewise, enhancers haven't evolved away these sequences, so they too are unstable. We think this is an alternative way of finding enhancers. It's very, very sensitive, and we think that unlike the chromatin marks that are used generally, we think this picks out some of the strongest enhancers, which some of the chromatin, some of the programs that use chromatin marks call in some enhancers, we think, promoters, and those are some of the strongest enhancers. So anyway, if you're interested in enhancers, just looking at that paper. Okay, now if we turn back to Drosophila, took this exit to look at mammalian transcription, but if we go back now to our old love, Drosophila, and we look at, apply the GRO-seq approach to it, and compare it to this ChIP-seq, this chromatin IP sequencing, done a beautiful experiment done by Karen Edelman's group, see that you can see where the polymerase is, and it piles up on, and it covers genes. By GRO-seq, you can see this similar pattern. There's actually a lot of reads here if you were to change the scale, but to keep everything on the same scale, we can see that. And you can see many cases of pause polymerase, and indeed, and you get orientation information as well. There are some genes that don't show detectable pausing, but the vast majority of them, 70% of genes that have any polymerase associated with them, show this pileup of polymerase at the just downstream of the initiations. But the promoters in Drosophila are not divergent, all right? So we have cases where we don't have cases where polymerases are going in both directions like you saw in mammals. And the reason we think is, it comes from the work of Jim Kannaga, where he's characterized the elements around the start sites of promoters and found that they typically, in Drosophila, there are many elements in the promoter that provide orientation information, and we think that that's perhaps the reason why we don't see divergent transcription in the fly. The fly straightens out the polymerase. Maybe it's a little more evolved than humans, I don't know. Okay, so we decided that it would be critical to understand the fate and properties of the pause polymerase to inform us about the mechanism. So we decided to take this head on and look at the kinetic fates of the pause polymerase and see if we can distinguish between whether there's anti-termination mechanisms or pause release as a mechanism by which polymerase now can be released into transcription. What is the regulatory mechanism? And so you can envision that the pause could escape, and we know it escapes, obviously, and you could be affecting this rate of this transition to this elongation state. Alternatively, we might have a situation where the pause you're forming the pause, it rapidly terminates, and the polymerase recycles, and if this is happening rapidly enough, you're going to get an accumulation of polymerase on the promoter region. So we decided to look at this both optically, see if we could do this both optically and biochemically. So we made, again, use of the polytene chromosomes, and this time we have a polytene nucleus where all the chromosomes are folded up, and we're using 2-photon laser scanning to optically dissect this. And you can see that you can get information on the banding pattern, and you can identify where genes are in this kind of microscopy. And if you do a heat shock and you have a polymerase that's GFP labeled. You can watch from non-heat shock to heat shock, the polymerase get recruited, and it's recruited to these pair of puffs that I've described to you early on that contain the HSP70 gene. So you can watch this recruitment of polymerase in real time. So, but there's a problem in looking at the polymerase that's there before heat shock, that one pause polymerase, and seeing that over the background. So to overcome that, we made a transgenic fly line that had an HSP70 gene that was marked by an M-cherry LAC-I, because this construct contains 256 copies of the LAC-O operator. So if you express a LAC-I with M-cherry on it, you're going to get a red gene here, and that's going to light up in the nucleus in a very specific way, is the idea. And we also introduced into this line a polymerase that was photoactivatable, and so it was photoactivatable GFP. And so we could find, the idea is you find this red dot in the nucleus, and then you can shine UV light on that to photoactivate the GFP, and now you turn that polymerase green, and you can watch that in a background that's fairly low. And then monitor the dynamics of that photoactivated pause polymerase. And so this was a challenging experiment, but we're focusing in on one part of the nucleus where the RFP LAC-I is located. We photoactivate that. This is the before photoactivation. After photoactivation, we get the band polymerase we're hoping to see, and we can then watch it decay. We can get the kinetics of loss of that. How fast does that turn over? And then we can go back and check that we really hit the right place by then doing a heat shock, a photoactivation and heat shock, and we can then see that this was indeed the puff region that contained the heat shock genes, and we're convinced that it's right. So from this kind of analysis, we determined that the pause polymerase is relatively stable, half-life of several minutes. We complemented this, and I am not going to go through all of this, but again, the paper is here, this is published, where we looked biochemically at the rates of escape and the rates of termination, and that agreed perfectly with the optical approaches they were doing, and then we looked at what happens in response to heat shock. What rate are we changing? And it turns out what we're changing is the elongation rate. We have very little effect on the rates of termination. So the heat shock affects escape, it doesn't work through an anti-termination mechanism. So that's where the effects are. And so who is doing that? Well, let me just say that was the HSP70 gene. You may want to see this in more than one gene, and you might want to see it in mammals as well. So we made use of a drug called triptolide, which blocks any initiation. So we can then watch the pause polymerase as it decays and then quantify it. And doing that in mouse ES cells, we can get rates with which that pause peak decays, and we can do this on many genes. In fact, we did it on 3,200 or so genes, and got rate constants for rates of loss of that pause polymerase, half-lives of the pause, and with nearly 3,200 genes. And they agreed very, very well with the Drosophila HSP70, several minutes in a range of half-lives. So the vast majority of these polymerases are stable and probably working like the HSP70 gene. And you can read more about that if you're interested. And there's a very nice work from Karen Edelman's lab on 13 different Drosophila genes where they measured also half-lives of between 2 and 20 minutes of other Drosophila genes. So we think this is pretty general. So how does the polymerase, how do we accelerate the polymerase escape? Well, the work of Dave Price suggested long ago, and others, that PTFB kinase might be working on to accelerate the escape of polymerase from its pause site, or the release from the pause site. So we decided to test whether one could, this was some years ago we did this, where we added GAL4 binding sites and introduced a fusion protein that had the GAL4 DNA binding domain fused to this kinase. So we could artificially recruit that kinase to target sequences and ask what happens to gene expression. And when we did that, we saw that the gene expression of HSP70, in that case that was so marked, was dramatically increased. It was nearly at the heat shock levels, transfection assay. We assayed the RNA by primary extension. I'm not going to, again, this is something that's been published. So the direct recruitment of PTEF-B is sufficient to activate HSP70 even without a heat shock. So without a heat shock, if you bring in PTEF-B artificially, you can trigger the expression. Now, how general is PTEF-B use in expression? Well, we know that 95% of the genes, probably all the genes, nearly all the genes anyway, depend on PTEF-B for expression. If we add a very specific inhibitor to PTEF-B, something called flavopiridol, it blocks the activity of that kinase, and we can then watch watch the genome, watching every gene if we, as polymerases clear after we've blocked the escape. The pause remains, but the genes get cleared of polymerase, and we can then get the rate with which these polymerases are also moving and get elongation rates. But the key point is that all genes show this pattern. So even genes that didn't show any pausing to begin with are left with a peak at the pause site and show this decrease, this dramatic decrease in expression, telling us that all genes have to go through this step. And this is just showing an experiment with flavopiridol, demonstrating this, that the peaks on if you block the escape by adding flavopiridol, you block this step, then the pause piles up. We see an increase. I just want to show you that there is an increase, and you can see the raw data showing an increase here, the red indicating an increase, the blue showing a decrease in the gene body. So this thing is the genome-wide view. The conclusions of this part is that Drosophila mammalian genes exhibit promoter proximal pausing. Most genes do, but promoter proximal pause is generally stable, not a consequence of rapid rounds of initiation and termination, but it goes out there, it's parked. It's not infinitely stable, so it's not some blockade. It won't be a blockade necessarily to replication. Heat shock genes of HSP70 increase, heat shock activation of 70 increases the rate of entry into productive elongation. It doesn't appear to be an anti-termination mechanism. And enhancers are built and initially transcribed like promoters, the mammalian genes especially, but you see this divergent transcription. I should point out in Drosophila there's just one orientation but on genes, but on enhancers they have that architecture of divergent transcription like mammalian genes. All active genes undergo a pause release step that is dependent on PTEF-B. So now I'd like to move to unpublished data and tell you a return to, we'll focus on the heat shock system in both in Drosophila and mouse, and we're learning different things from the two studies. Well, they're largely overlapping. We're getting many of the same results. But what I'm going to do is highlight those studies from Drosophila that I think are richest in providing us information in promoter proximal formation of the promoter proximal pause polymerase, at least the allowing that step to happen, and then use the mouse studies to focus in on where HSF is working genome-wide on transcription. And so we'll be getting at these different rates of forming the pause and escape from the pause. So we know that NELF and DSIF are known to stabilize the pause, but there are other factors that are part of the core promoter that may contribute to generating the pause, to allowing the pause, the polymerase to enter and create the pause in the first place. Okay, so now I'm going to just turn to yet one more run on new technology, which is something we call PRO-seq, which is just like GRO-seq, except it gives us base pair resolution. So PRO-seq is like GRO-seq, but PRO-seq uses a biotin affinity tag, biotin NTP, whereas GRO-seq uses bromo-U NTP. And PRO-seq provides base pair resolution, whereas GRO-seq was only about 50 base pair or so resolution. The way this works is that the biotin, when you provide only biotin NTPs, a biotin gets incorporated. A second biotin incorporation appears to happen very, very slowly. And we mostly just get one base put in. Now we have an RNA that is extended by one base, and it has an affinity tag, this biotin NTP on. And now you can fish that out. And we do these 3 successive affinity purifications. And in spike-in controls with biotin and non-biotin RNAs, we estimate there's a millionfold purification. So everything you sequence is really signal. And everything is signal and not noise. So it's a very sensitive and economical use of sequencing. We then do, again, the ligation of the linkers and reverse transcribe ligation of linkers, and then do high-throughput sequencing on all of this. And what are the advantages of PRO-seq? Well, Ruanons in general give us this genome-wide distribution of transcriptionally engaged polymerases. So you can get snapshots of the kinetics of changes in transcription. It's not affected by the background levels of accumulated RNA, unlike RNA-seq, where if you're looking at repression, the RNA has to decay before you see a change in expression. This looks directly at the gene itself, so you can look at repression and activation changes that happen instantly. It provides base pair resolution as mapping where polymerases are. It's highly sensitive, orders of magnitude more sensitive than chip polymerase assays, and it's strand specific. So Fabiana Duarte, another grad student, did the bulk of this work where she examined Drosophila S2 cells, some help Mike Gerton, and so the protocol is simply take Drosophila cells, heat shock them, take time points at 30 seconds, 2 minutes, 5 minutes, so on, and then do PRO-seq and ask how does the polymerase distribution change in this kinetic time course. And you can then see those changes genome-wide. You can plot this out on what was called an MA plot, where we're looking at the ratio of heat shock to non-heat shock reads on the gene body. We're looking at the fold change in reads on the gene body as a function of time after heat shock. Each of these dots represents the reads on a particular gene, and we're spreading this out basically in the x-axis by looking at the average gene body counts. We then assess what's a significant change using the program DE-seq2, which uses the variation that we have in our replicates to then identify significantly up, pink regulated, or down, blue, up is regulated, pink, down regulated in blue. There are hundreds. We can see that there's very little change early on, what we hope, expect. 2 minutes, 5 minutes, we're beginning to see changes, and we see lots of changes by 10 and 20 minutes. Very fast response transcriptionally. Hundreds of genes are turned up. Thousands of genes are turned down. Okay, so this is a massive response. Whenever you see this kind of response, you have to really worry about how you normalize. We worried intensely about all of this, and I'll be happy to discuss that if anybody's interested. This is true when we look at the mouse as well. Okay, so here's a typical upregulated gene. We have pause polymerase, and then we see increases in transcription. Downregulated, we see decreases in transcription with rapid time frame. So we know major heat shock genes show this pausing. We were interested in the level of pausing more broadly on all of these activated genes, and how this might influence heat shock activation. And so looking at the average number of PRO-seq reads as the amount of polymerase in various parts along the gene, plotting as a function of plotting relative to the TSS, we see that the activated class of genes, those hundreds of genes that are activated, show very strong pausing, very high accumulation of polymerase. Again, in the same place we've seen it for Drosophila HSP70 at 20 to 50 bases. And some pausing, but much less in the unregulated and repressed genes. Now, we screened the modding code, there's these huge data sets that exist that have been generated by NIH-supported efforts of all factors and where they're located, and there are other publicly available databases. So we search these for transcription factor binding factors that are differentially enriched in the heat shock activated relative to the repressed or unregulated genes. And what we found from that was this factor that we had seen before, GAGA factor. As you may recall, GAGA factor was present on heat shock genes, as I mentioned in my early slide showing what we knew about GAGA factor from focus gene studies. Moreover, we know that mutations in the proximal GAGA element of HSP70 disrupts pausing. We looked in transgenic lines many years ago. Gilmore lab and Levine lab have correlated GAGA elements with pause genes. You've seen that too. And GAGA factor may have another role. It can interact, as shown by Dave Gilmore in vitro, with this NELF, this pausing complex, and may help to recruit NELF as well. We're going to be talking about a different kind of role of GAGA factor today. So let me back. So if we look at where GAGA factor is binding, this is what we call a cumulative frequency of GAGA factor bound genes. So we're looking at genes that have GAGA factor and where that binding is taking place as a function of distance from the start site. We're seeing that GAGA factor is binding within 100 bases. This is a log scale, okay? So bulk of the GAGA factor. And it's occupying a lot of the activated genes, and much more so than the unregulated or repressed genes, where the GAGA, you have to go much further out from the start site to begin finding these GAGA factor binding sites. And indeed, if we plot the total ChIP-seq density, you see a lot of GAGA factor associated very, very close to the promoter. The bulk of it is within 50 bases upstream of where the start sites are. So the GAGA factor seems to like to be in a particular location. Okay, so 75% of the activated genes are GAF bound. I kid that, if we had done this experiment first, we would have called GAGA factor heat shock factor, and this heat shock factor is only on 20% of these. So is this pausing that we see that's created by GAGA factor related to the heat shock activation? And to test that, we knocked down GAGA factor and then did PRO-seq and with a control, a LAC-I knockdown. And indeed, the knockdowns work by this Western, and we can then see all GAF-bound genes shown in purple, except those that have also significantly changed, which they're only significantly changed during green, but you can see there are a lot of red genes, red dots here representing genes that are both have GAGA factor bound and are significantly affected by the GAGA factor knockdown. We're looking at the effect of GAGA factor versus a control RNA on gene body count, so after 20 minutes of heat shock. So GAGA factor matters on a subset of genes. There are some GAGA factor bound genes that it doesn't matter. It matters on about 20% of the activated genes and only about less than 1% of the repressed genes, so it doesn't have any role in repression. If we now look at these red genes that are, when you knock down GAGA factor, you see a decrease in decrease in the ability to activate these genes in response to heat shock. You see a decrease in the level of pausing, the amount of PRO-seq reads on the pause site. Whereas if you look at this other class that seems to be unresponsive to the GAGA factor knockdown, even though on these genes GAGA factor is reduced, we don't see a change in the expression. There the pausing hasn't changed. So we see this positive correlation with genes that are dependent upon GAGA factor for heat shock in terms of the amount of pausing, the effect reduction of pausing, and we don't see that. We see an anti-correlation with regard to dependent genes. This is just a box plot showing result. We can look at individual genes just to reassure ourselves that we're looking at the right thing. Here is a gene going right to left. Here's the pause polymerase on before heat shock in the control, and after the GAF knockdown, we lose that pause polymerase. We now look at heat shock On the control, we see an induction of the gene, and we don't. And we see a dramatic reduction in expression of that gene upon heat shock. So both pausing and expression go down in concert. So we screened on 92 different transcription factors and histone modifications to try to understand what are the factors that can substitute for GAGA factor in pausing. Because remember, there are all these genes that show pausing, but they're independent of that pausing is independent of a GAF knockdown, and their activation seems independent of a GAF knockdown. And so when we did that, we found two proteins, well, a couple of proteins. One is M1BP, another core promoter element factor, factor very, very close to core promoter, was discovered by Dave Gilmore, and then we also saw insulator factors, which are often associated with promoters or near to promoters, beef and chromator, which has multiple activities, but it also shows up as with insulators, and it probably has an insulator activity. And we then correlate, if we look now where M1BP is in the class of genes that are dependent upon GAF and those that are not, we find it is dependent. We find that the M1BP is present in the GAF independent, but not the GAF dependent. Likewise for beef and chromator. And so this is just a Venn diagram showing that GAF dependent heat shock activation genes do not overlap with those genes that have these insulators or the M1BP. So there are multiple ways of creating pause polymerase. How is GAF working to allow this pausing? Well, we know that work from Carl Wu had shown that GAF interacts with nucleosome remodelers to keep promoters open. And so GAF binding might just open up chromatin, and that would allow polymerases to enter. And to see then genome-wide, if this might explain all the data that we have, we did a micrococcal nuclease. It was work of Nick Fuda, and mapped all the nucleosomes genome-wide under various conditions. And so we just digest micrococcal nuclease, isolate the mononucleosomes, do paired-end sequence, and now you know where, map those to the genome, and then you know where every nucleosome is under various conditions. And so we compared the condition of the unaffected genes, those where GAGA factor is knocked down, nothing happens, and those where we have reduced pausing. And in both of those cases, the promoter is largely free of nucleosomes. This is a common feature of promoters. Nucleosomes tend not to occupy the promoter regions. They tend to be open. Now, what happens when we knock down GAF? We see, in particular, I'm going to focus on those that show where GAGA factor knockdown causes reduced pausing. On those genes, shown here in red, we see a filling in of the promoter with nucleosomes. It looks as though GAGA factor has a role in keeping the promoter open and ready and accessible. So GAF could be working directly with remodelers to open up chromatin structure. We also wanted to see if we could rule out the idea that maybe it's GAF creating the pause polymerase, which in turn opens up chromatin, as was shown by as a model that was supported by data from Karen Edelman's lab very nicely. But we know that pause polymerase, if you knock that down by knocking down NELF, that you can get filling in of many nucleosome sites. But we wanted to test whether GAF may have this activity on its, even without the presence of polymerase. And so we examined GAF binding sites that were far away from promoters, that were not associated with promoters. We looked at 115 intergenic sites that are bound by GAF, and you saw the same results, where knocking down GAF leads to filling in of these regions that were nucleosome free with nucleosomes. So GAF increases nucleosome RNAi knockdown, increases nucleosome occupancy at binding sites that have no detectable pause polymerase. There is this direct. So here are the conclusions. Part 2, GAF contributes to creating proximal pausing. GAF is essential for the activation of genes with GAF dependent pausing, and so we envision that we have binding that allows GAF that allows pause polymerase, and then upon heat shock we can induce the gene and get activity, but if you have a GAF depletion on the subset, you don't induce the gene. You don't form pause polymerase. Insulators and M1BP provide an alternative to GAF generating pausing, and that these tend to work these tend to occupy in a mutually exclusive way. And the GAF is critical for establishing these nucleosome-free regions that allow transcription factor access and the formation of pause. For the last bit, I'm going to try and go through pretty quickly. Hang with me. These mouse studies, which evaluate mechanisms of HSF1. So HSF1 is, I mentioned, the master regulatory protein, right? That's what everybody says. That's what we all said, So we were a little surprised by the results that we got here when we started looking at HSF1 mutants. Now, just to give you a little background on HSF1, showing you that it's interesting beyond heat shock. It has contrasting roles in cancer and neurodegenerative disorders. Susan Lindquist's lab showed very nicely that in an HSF1 minus mouse, you knocked out HSF1. The mice aren't happy if you heat shock them, obviously. They'll die. But without a heat shock, they're fine. And in fact, if you paint carcinogens on their back, the wild type, of course, will develop tumors. The mutant doesn't, or it's much delayed and many fewer. So HSF1 seems to be an enabling cancer. I mean, it's not too surprising. Cancer cells have high demands, and they're stressed in a sense, so they need the stress response. HSF1 also drives chaperone protein synthesis, and people have been talking about using overproduction of HSF1 or chaperones to treat folding disorders. So in one case, HSF1 is bad. It helps cancers grow. In the other case, you may want it for preventing some of these neurological disorders. So we thought it would be, in addition to using it as a model for studying transcription, it would be useful to try to dissect more completely the heat shock response and the role of HSF if one wants to think about developing therapies down the road. So we made use of a wild type, mouse embryonic fibroblasts, and an HSF1 minus fibroblast line. We obtained Benjamin lab. And we then took these two different strains, wild type and mutant, looked at non-heat shock, and then did again a time course with PRO-seq assays. And I'm going to show you that the response is a little bit complicated, or more, I should say, sophisticated, and that it's not simply that every gene behaves like HSP70. There are many genes that are activated by heat shock in a time course. Go through these. See activation, strong activation by 12 minutes here of this particular classic heat shock gene. And in the mutant, there's no induction. So that's good. These are genes that are dependent upon HSF1. There are many activated genes that look like this, and these tend to be cytoskeletal genes where you activate, you can see the activation is rapid. I mean, you can see in 2.5 minutes we're getting induction. By 12 minutes, a lot of induction. By 60 minutes, it's back off. So it's a transient but rapid induction. And if you notice, in the mutant, we look at that same time course, it's independent of HSF1. So HSF1 isn't quite as much of a master regulatory protein as maybe we thought. So there are genes that are activated of HSF1 as rapidly as the classic heat shock genes. Some genes are induced late, and those are HSF1 independent as well. And then there are thousands of genes I mentioned that are repressed, and that repression is also heat shock factor 1 independent. So HSF1 is not involved in the mechanism of repression, we think. And so if we look, and again, at these MA plots, looking at the changes, again, the yellow is activated and blue is repressed. There are, again, many hundreds of genes, close to 1,000 mammals that are activated and many thousands that are repressed. And if you look in the mutant, there's a similar number that are activated and repressed. So we're interested in what are the overlaps between the wild type and the mutant. And for activation, there's some overlap. There are some genes that are clearly expressed only in the wild types. They're HSF1 dependent. But in terms of the downregulation, the overlap is much more complete, and it looks like there's very little role of HSF1 in this massive amount of repression one sees. So what is the distribution of the HSF1 protein? So we can look at that by, again, doing ChIP-seq. We had made a couple different antibodies and looked at the time course of HSF1 binding, and we saw a couple different types of kinetics. And so we can map, we have all these positions. And for major heat shock genes, the HSF1 likes to bind very, very close to the promoter. But if we look at all activated, all the HSF1 peaks, and we do a cumulative fraction, we find that many of the binding sites are far from promoters. But the density is highest, of course, in the promoter region. So we can then ask what happens in these class of genes where we see heat shock-induced HSF1 binding close to the promoter, within a kb of the promoter. And these are just showing the binding changes for this large collection of genes. And we can then ask, how does PRO-seq density change, both in wild type and mutant? We can find that there are genes that are HSF1 dependent. That is, they are induced in wild type, and the red is indicating fold change increases, but not in the mutant. Here, these are expressed early and late, and anything reduced. There are other genes that are independent of HSF1, shown in red, where we see induction in both wild type and mutant. Here we see induction in these early time points, both as well. And there are some genes where we have HSF1 bound close to the promoter, but there's no effect on transcription whatsoever. What is the mechanism by which HSF1 mediates this activation? One could envision that it could be working at either these two steps, the entry or the escape. How does HSF1 matter? We know that there's an increase in transcription upon heat shock. If we look now at, again, these genes that are HSF1 dependent, and we look at the change, fold change on the gene body, in wild type we see genes are all going up. And of course the mutant, they're not changed or going down slightly. And if we now look at the pause region, they go up in both cases, and these levels are independent of HSF1. That is, the HSF1 minus minus do not affect the levels at the pause site. So we're still getting polymerases entering the promoter, and they're actually increasing. We think that increase might be due to the fact that the thousands of genes have been repressed, and so there's just much more polymerase, and perhaps it's just filling in by mass action. But the step that's dependent upon HSF1 is this escape into productive elongation. Okay, and so, and there are some HSF1 that isn't bound on promoters, but when we knock down HSF1, those genes, there are sets of genes that are activated, and so we assume that these might be working as enhancers. And if we look at those in a similar way, the change is in the gene body, but not in the promoter box plots. So it's arguing that both the cases where HSF1 is bound or close by or enhancers, that it's acting at the step of release of the pause into productive elongation. So what about repression? What is the role of HSF1 in downregulated genes? I already mentioned there's probably very little role. There are many active genes are significantly downregulated. They fall into two classes. There's a set of genes that are turned down pretty rapidly, early downregulated genes, and there's others that are late downregulated. And it may be just some general mechanism, except that these genes fall into specific classes. The early downregulated genes are shown in brown, and both downregulation happens both in wild type and HSF1 minus minus, so it's independent of HSF1. The early genes, shown in brown here, are highly enriched for genes involved in metabolism and cell cycle. You want to shut down most of the cellular functions. The blue genes, this later late downregulated are separate classes of genes. These are involved in RNA processing, mRNA splicing kinds of events. So there's probably it's probably just not a general inactivation of some component here. It seems to be somewhat regulated in that these fall into different classes of genes. It's just the data showing what the heat map looks like for the density of polymerase before and after heat shock. You see this downregulation, decrease in green, no change actually around the pause. Again, just the difference, fold change. We see decrease on gene body, but if anything, maybe a slight increase, no change on the pause region, and that's also independent of HSF1. So we think that there are multiple kinetic classes of transcription regulation upon heat shock. Remember, there are these steps of recruitment, and then we quickly go to the pause site. There's never detect, rank Pew by ChIP-seq has never detect piling up of polymerases by ChIP anyway at the start site, even though he sees TBP and 2B there. It seems like polymerase, once it enters, goes quickly to the pause. And we can then ask, how is recruitment and release affected when we look at these various classes of genes? There's the class that is induced, and this yellow shows the fold change. Here's the scale. We see changes as early as 2.5 minutes with this class. These are the classic heat shock genes. And we see that the levels go up rather dramatically. And the effect is at the level of pause escape. We see an increase in the pause, but it's independent of HSF, and these are the HSF-dependent genes. There are another set of genes that are as rapidly induced, but they're induced in a transient fashion, and they're independent of HSF. And in fact, these genes are cytoskeletal genes, and if we do a search of what are the elements there are elements that are bound by serum response factor. And serum response factor is interesting because it's regulated by a free actin it senses the state of the cytoskeleton. And so it makes some sense that these cytoskeletal genes might be responding. We know that upon heat shock, the cytoskeletal changes dramatically, and so maybe that collapse or change in cytoskeleton due to the heat shock is somehow triggering serum response factor to activate these other genes in a transient way. There's some genes that turn on late, and there are two classes of genes that are repressed, and they both seem to be repressed at the stage of escaping from the pause polymerase, at least in mammals. So just a couple of cartoons at the end here, just to show you that GAGA factor we think has a role in, or I hope I've shown you that GAGA factor has a role in keeping nucleosomes off of promoters. It also helps formation of the pause, which we know from Karen Nadelman's work also helps keep nucleosomes off of promoters and available for factor entry and polymerase entry. This may also be important in allowing for the synchronous activation of regulatory genes that are seen by Mike Levine in the early embryo. Genes, hundreds of genes that specify pattern in the embryo are turned on in a very precise way in a very short time window, and this is and this rapid response and complete response depends on pausing, as shown by Gene Lab. We can think also about this pausing as providing another way of synergizing with the recruitment. That is, there are factors we know are important for getting polymerase onto the promoter, like GAGA factor, but then there are factors like heat shock factor that have a role in the escape of polymerase. And the two together we envision, can by synergy provide us a very dynamic, large dynamic range of regulation and the ability to have two different input signals be contributing to regulation. And finally, polymerase is going through this stage. Polymerase is undergoing many, many changes, and perhaps this provides us, that pause provides us a point for picking up the RNA processing factors that we know decorate the polymerase and follow it as an entourage. So I just want to thank the people in my lab who, here we all are in the same boat, and there are certain people who earned halos for this presentation. I as I pointed out days ago, I don't want to say that these other people don't deserve halos. They do, but they're doing different things, not related here. In particular, I want to thank Fabiana Duarte for the Drosophila unpublished Drosophila work, and Jay Mahat for the unpublished Mammalian work on heat shock factor, and also Nick Fuda for his work on the GAGA factor in particular. So there's still room in the boat if anybody wants to come on board. We've thrown a few of these people overboard. No, I'm only kidding. We didn't really throw them overboard. They went on to better boats, bigger, better boats. So let me just leave the conclusions up here, but let me just pick up. I talked about many of these before. But you know, the heat shock response. Let me just pick up with the new conclusions that causes rapid and large-scale diverse changes in transcription. The classic induced heat shock proteins and others are absolutely dependent upon HSF1. HSF1 at promoters enhancers regulates transcriptional activation of genes at the pause release step, not at POL2 recruitment. And the cytoskeleton genes, this class 2, are activated as early as the classical HSP genes in an HSF1 independent way. We think that's working through SRF, serum response factor, which is picking up the cytoskeletal changes we've seen on heat shock. HSF1 does not mediate heat shock-induced transcription repression. Repression seems to be independent of HSF1, and the massive heat shock downregulation in mammals is also at the pause release step. We're getting less pause release. Okay, with that, I'll take any questions you might have.
That was a wonderful topic, thank you. So, especially for the last bit with the HSF1 kinetics part, how does epigenomic or epigenetic modifications like histone modifications or in mammals DNA modifications fit into that? You showed that the various classes that in terms of the sequence context that matter, like there's difference.
Right, yeah, so yeah, we haven't, yes, so we haven't probably looked at that enough. You'd probably be interested in both the histone modifications and status before and after, right? Yeah, and we've of course spent some time with looking at HSP70 over the years and found that it gets modified in the modifications of the histone modifications. Active marks get put on pretty rapidly, and they also seem to go. And gene returns to the normal state. But we haven't looked at those kinetics probably as closely as you may want to see. But basically, the marks sort of change with the expected marks come with gene activation.
Right, but I'm kind of more interested in when it's repressed.
Oh, when it's repressed. Oh yeah, so the repression of the thousands of genes is still mysterious. I mean, we don't really know how that's working. It seems to be working on it. I mean, we still have pausing taking place. Polymerase seems to still be entering, at least in mammals. That's the one place that's a little different in Drosophila. But in mammals, we see this, and the escape seems to be limited. And but we haven't really looked at what is changing in terms of could it be a chromatin kind of shutdown? Yeah. That would be an interesting point.
Thanks.
A couple of related questions. Why do you think cells use promoter pausing and clearance as the main way of regulating transcription, as opposed to maybe an early step like promoter recognition?
Well, I don't want to say it's the only way they regulate. I mean, well, there's a lot of pausing. Genes go through this pausing step. In some cases, it's rate limiting, and in many cases, we've seen that there is some regulation of that step. So why does it do that? Well, it does provide an opportunity for a second set of inputs. You know, as I tried to illustrate in one of the cartoons, it wasn't a terribly good illustration, but you have the ability to change the entry rate, which gives us a chance to regulate the early steps, but you can also regulate at this later step, so you can have two different kinds of signal inputs to give a little more complexity and sophistication to the regulation. Plus, you can also synergize from those two and get probably higher levels of expression as well, as we know. if
Of
you
course,
get two
that
steps.
would be good if this is two different ways. But is it true from
Yeah.
what we heard that it's actually the major one, not the
Yeah.
parallel one compared to the first?
Yeah, a lot of the entry steps seem to be, that I've described here anyway, are largely constitutive factors that are there, you're right. And so, well, one obvious advantage is it's a way of keeping the promoter open and accessible. So if you have responsive genes that have to go up and down, you know, if you have the polymerase there and an open. Yeah, and I think when you have mammals, you know, you have the two polymerases like that, you really are keeping this region open. You're also preventing, a polymerase is a good barrier to propagation of repression domains. So I think maybe in Drosophila the genes are close enough that you don't really need to build these pairs of genes to keep repression that may be happening far upstream and propagating down chromatin from entering promoters. But in mammals where those distances are large, you have plenty of places to nucleate a repression event. Putting pairs may have some advantage. So the pause polymerase may have some role in that, and a byproduct is that then you can modulate at that step.
Okay, related question actually came from undergraduate students when I talk about this model.
Yes.
Why do you think after opening pausing you make a small RNA? Why does this small RNA has to be aborted in a cycle of abortion?
Oh, I don't think it is. It isn't. We don't. Well, we don't. Unfortunately, we don't know for absolute sure what that. I mean, those are really hard experiments to ask. Does that polymerase simply extend on that RNA? I mean, certainly it's extendable. We know in nuclear run-ons, in nuclear run-ons in vitro, that it's extendable. We know that we have pausing even when we're firing rapidly. So if you were to just generate that RNA, just to have it be aborted, so that you then do another one, and in the case of heat shock, we still have pausing. Even when we've activated the gene, you would have to every 4 or 5 seconds be making an RNA that you abort just to replace it with another one. I don't think that's likely. I think, and we're trying to do these experiments that would actually address that question directly. But my hunch is that polymerase that is pause is capable of extending.
In an induced state, is there an abortion of the short RNA? That's the question. Or if it is,
well, we don't detect that RNA certainly. As a much of a free RNA. There's not much of that there, so I don't think it is, and it certainly doesn't go up upon heat shock. I mean, there are a lot of reasons why that hypothesis is unlikely, but I can't say we've definitively ruled it out in all cases.
Even in the seminar earlier today, the model has that as well.
Oh well, no, there is an abortive. Okay, I'm sorry. Yeah, there is an abortive kind of transcription very early on, but once you escape, once you go through this thing called promoter escape, I probably shouldn't use the word escape, I should be using pause release. Once you go through that promoter escape, then you enter the pause. I don't think you're not aborting very frequently. That before the clear. Yeah, no, there. I mean, yeah, you. Well, why do they want to do that then? Oh. Somebody want to answer that? Who knows more than me about that? I got some head shaking no back there. Not clear why they want to do that. Beth, you want to take that? Yeah. Why do we have abortive initiation? You know, before you.
I think it's my own view. We don't really know, but my own view is that it's built into the.
It's built into the
That's the way that it works, and that's what happens.
The only way I can answer my student is there's no other way to keep it there for too long. So I have to let go and then synthesize again and let go. Because it's natural decay and no way to keep it. That's the only way I can imagine to answer the student. That's why I'm asking the experts here.
I wish we could give you a better answer.
That requires some beer.
So wine, maybe, would that help with the discussion outside? Some wine and reception outside?
I'm game.
Okay, all right, let's thank John.